I am Sarah Bugami, and I will talk today about applications of fluorescence in situ hybridization. The fish is used in detection of chromosomal abnormalities, a technique in which single-stranded nucleic acids are interacted so that hybrids are formed by molecules with similar sequences, enable us to assay multiple visualized, localized signals in a single specimen, gives power to resolve several genetic elements, multiple gene expression patterns through multicolor visual display function, help to a particular where identify within an individual false gene chromosome. The types of fish probes: whole chromosome painting probes, repetitive sequence probes, locus specific probes. Painting probes, whole chromosome, complex DNA probes derived from a single chromosome, most useful for clarifying chromosome rearrangement in metaphase. Example: genetic translocation in acute lymphoblastic leukemia, available for every human chromosome, led to development of two independent fish techniques: multicolor fish, M-fish, and spectral karyotyping. Sequence probe repetitive, hybridized to specific chromosome regions. Examples: pantelomeric probes. Satellite DNA probes hybridized to multiple copies, resulting in a two very bright fluorescent signals, metaphase and interphase, suitable for detection of monosomy, trisomy, etc. in both leukemias and solid tumors. Probe locus specific is to detect structural rearrangements, bind to gene of interest to detect amplification and deletion. Using multicolor fish, recurring translocation can be identified. Examples: PCR. PCR gene detected with green and red fluorochrome will appear yellow spot in leukemia cell. Principles: basic element, a DNA probe and a target sequence. Before hybridization, the DNA probe is labeled by various means. Two labeling strategies are commonly used: indirect labeling and direct labeling. The labeled probe and the target DNA are denatured. Combining denatured probe and target allows the annealing of complementary DNA sequences. If the probe has been labeled indirectly, extra step is required for visualization of the non-fluorescent hapten that uses an enzymatic or immunological detection system. And this is a case study and examples for diagnostic application of fish of a 4-year-old girl who was referred to a laboratory to investigation for clinical obesity, mental deficiency, and respiratory problems. The patient was born for non-consanguineous and healthy biological parents. And for detecting etiology, fluorescence in situ hybridization using probes for small nuclear polypeptide N, SNRBN, was necessary to confirm the deletion of paternal chromosome 15, which is the predominant genetic defect in Prader-Willi syndrome. Diagnostic application of fish: prenatal diagnosis, cancer diagnosis, molecular cytogenic of birth defect and mental retardation, the identification of specific chromosome abnormalities, the characterization of marker chromosome, interphase fish for specific abnormalities in cases of failed cytogenetic, monitoring the success of bone marrow transplantation. Thank you for listening.